invitation for organizing this great event. So, the Allen Institute traditionally been well known for making large reference data sets, which we make available to, which we make for the neuroscience community in the hope of driving, understanding of the brain forward. Given that this conference is about neuroscience and data science, I thought that I'll talk about the making of one of these large-scale data sets, which we call an atlas. So hence the title, Constructing a Mesoscale Mouse-Brain Connectome, and the making of the Allen Mouse-Brain Connectivity Atlas. Okay. So the goal of this project is to map out in mesoscale all the connections in the adult mouse brain. And we're going to get at this information by using a virus. You inject the virus into the brain, and then this virus mediates a green fluorescent protein, which means that all the infected cell, effectively, it glows, and all its processes and all its axon glows this green color. And we have this fantastic imaging platform that allows us to collect high-resolution images, but at the same time, it keeps the pristine 3D context of the data. So that means at the end, we can see the axons, and we can also trace the axons throughout the brain. So then for every single brain that we've done an injection, we end up with 140 coronal serial sections. Each sections are 100 microns apart. And in one image, the pixel resolution is 0.35 microns. And you get an image that's approximately 40,000 by 30,000 K. So the whole scope of the project is to make 2,000 data sets that are just like this. And the other thing that the Institute is famous for is our genetic tools. So these are special transgenic mice that when we inject into them, it will only infect a subpopulation of the cells there. So a combination of wild-type mice, we're injecting it all over the brain, and these Cree lines, transgenic mice. So this is where the data science part comes in, because we're going to end up with 2,000 experiments that yields 250,000 images, is about a petabyte of data. So then how do we convert all this data so that we can organize it, browse through it, extract meaningful biological information out of it? And the key to that is then converting these RGB pixels from our images to something biologically meaningful. So we created this data processing pipeline. First thing you need to do, where are the axons in the images? That's our signal detection. Each brain is slightly different, and it's put into the machine in a slightly different angle. So you need to take all of that image data and put it in a common coordinate system so you can compare between the images. And also, I need to cast all this data to some classical anatomical atlas so that people can then integrate that information to the information that's already known in the literature. So within 15 minutes, we probably won't be able to go down the details of all the algorithms. I'm just going to talk about some of the challenges that we had while trying to process this data. The first step of the processing is to do signal detection. Effectively, we want to find the difference, identify all the pixels that are signal positive versus the background. The challenges for segmentation is always signal to noise. It's always, we want more signal, less noise. You can see there's per tile inhomogeneity. The fact that this is orange means that there's high background in the green channel as well. So, and then this background changes depending on what part of the brain you're in. So you just can't do simple thresholding. Also, the variety of what we call positive signal, there's a large variety. It ranges from these high, bright axonal fiber bundles where all the axons are traveling together. to these dense terminal fields where there's connection, strong connection from the injection site to this area to brain, and also to these single fibers. So while when we describe our paper, we talk about the segmentation algorithm is effectively multiple segmentation algorithms that have been applied and then joined together to get to make a single segmentation mask. So, as I said, to make the data useful and to provide anatomical context, we need to map all of this data to a common space and also relate it to classical anatomy. So we built a 3D reference atlas from a very traditional 2D reference atlas. So the Allen reference atlas was done in a very traditional way. Brain was sliced and then stained for nissels. So a nissel stain basically show every cell as these tiny purple dots. And when you see differences between the density in the dot, that will signal to anatomists that is there's a different area here. And then traditional 2D drawings are done. Because we needed a 3D space, we had to take all the nissel sections and then reconstruct that in 3D. Because we're looking at connectivity, and there's lots of connectivity going from one hemisphere to another, we needed basically a symmetric 3D space that's annotated to both sides. So what we did was then we flipped the reconstruction through the middle, and then we extrapolated all the structure, and then pushed it. We then had to do some interpolation and pushed it all into 3D space. So then once we have a 3D space, the idea is that we want to map all our connectivity data, and then in previous projects, we also mapped gene expression data to this same space. Once everything is mapped in the same space, you're able to compare data sets within a data modality, across data modalities, and then through our API, you can grab the data and then register it to other data sets as well. So for this particular project, the challenge with registration is that our specimen data, from this 2-photon tomography, is very different to our nissel data. And when you try to do that type of registration, there are methods to do multimodality registration, but if you make your cost function very complicated, then it's not very robust. You can get stuck in local minimas very easily. So what we wanted to do was introduce introduce a template in the middle that has a similar profile, to each specimen data, so that this optimization problem is easy, and we can do that robustly many times over, and then we'll take this as a one-off problem, which we can spend more time, with manual intervention and doing more tweaking. So at the beginning, we thought, well, we could take one particular brain as our template, but then the problem is that none of the data sets is perfect. There's no perfect brain. And then if you pick a bad one, then all your registration would, you will cause biases in there, and you will lock into all the peculiarity of that one particular brain. So then we said, okay, let's make an average population template. That's been done a lot in the MRI community as well. So the problem with trying to make an average is that you need a good registration algorithm to make a good average, but then you need a good average to make a good registration algorithm or registration protocol. So our solution to that was to actually do it iteratively. You pick, in fact, we took a subset of brains and then just try to register it to the nissel, just using affine, because there are less parameters. You get stuck in local minimas less. And then you get a rough average. And then now you register everything into this rough average. And then as you iterate over, everything becomes more clear, especially after you introduce the formal registration. So this average template is actually averaged over 1,200 specimens. And that you can actually see very fine level details that you get from a single specimen. So while we were looking for connection in the brain, this average template actually tells its own anatomical story. So earlier on, we heard about that the mouse uses whiskers to find its way through a maze or how it navigates. So you can get imprints of these whiskers throughout the brain. So that's in the barrel cortex. That was the part that was being optogenetically moderated or silenced. So each one of these little blobs here in the barrel actually correspond to one of the major whiskers on the mouse. And you can actually see it on the brain. You can sort of just see it in one particular specimen, but when you register a population together, they stand out very clearly. Even more interesting was this is now at the very back of the brain. So when we first saw this in the template, our guy who did registration, he says, "What's that spirally stuff at the back?" So these are actually another. These are again represents the barrels in the whiskers. So each one of these little segments again represent the one of the major whiskers in the mouse. So while we were looking for connection, we also found this beautiful anatomical template that we can use for registration. And then moving forward, we're actually going to change our common coordinate system to this, because this is now beautifully detailed and it's also smooth. And when you're doing registration, you want to register smooth things so that you can converge more easily. All right. The next step that we take with the data is that we do this gridding or voxelization. So after we've done our segmentation, you get a mask like this. And then for every 100 micron little block here, we're going to compute a local statistic. For example, we compute the projection density in each one of these blocks. And then because we're sampling every 100 micron apart, we make 100 micron isotropic voxels. And then we're going to then resample this entire grid back to the 3D coordinate system. So then you'll end up with, for each experiment, you end up with 500,000 voxel. That is a lower dimensional description of your entire data set. The reason why we do that is that at this resolution, we can actually read our entire data set into memory, and we're able to offer a lot of on-the-fly searches for our website. And also, people can download this data so that they can do actually a whole data set analysis. And I'll show you some of those a little later. So this is an example of a visualization of the gridded data in which there's injection into the motor cortex. And then you can see, and remember that when we do the injection of virus, we're looking at its axonal processes. So some cells in the motor cortex actually have axon going all the way to the back of the brain. So now that we have done all these processing, how do we use it now to organize our data and to present the data? So because everything is in the same 3D space, we can actually give people a quick glance of where all the experiments are. This was actually from our previous release, in which we had 1,772 experiments. We can overlay this on a 3D map. Each of these circles represent one injection. The size of the circle represent how large the injection was. The colors represent one of the colors of the reference atlas, so it gives an indication of where that structure is. And then so you can explore this data set by clicking on one of the circles. And when you do that, we show a preview of the data over here. So here is like a standard Google Maps zoom and pan. So you can access the entire image series just right here. Here we have now the MIP again, the maximum intensity projection of that data. We've pre-cached a whole bunch of views so that you can actually spin it around in the horizontal axes and the vertical axes, so you can have some 3D visualization of the data. And then these two views are synced, in which when you click on one of the big pathways here, the 2D images would update. While there's a lot of data sets here, you can also filter by your region of interest or by the tree line of interest. So you can also click on this I button, and then we also recast the data as a histogram. Basically now, because we register all the grid into that 3D reference atlas, now you can actually say how much proportion of the signal that from that whole brain is in the different compartments of the brain. Again, this view is also interactive with these two views. That's because algorithms are never perfect, and when you see a particular value here, I think it's important to actually show people where the primary data is so that they can always confirm what's going on in this view. So the images I'm showing are always shown in these small little windows. You can always expand it out so that you can view your data in a full page, or you can go to a 3D view of the data. So in our 3D visualization, what we do is that with our gridded data, we actually try to, we do virtual tractography, in which starting from every voxel, we trace the data through the gridded data, through the signal back to the injection site. So then for every data set, we can build a skeleton of the projection pattern that you see. And once you build a skeleton and turn it into these graphical objects, it is easier to overlay a lot of experiments together. So here we can overlay 21 different experiments because you can just give them a color. And also once you've done that decomposition, you can also turn lines on and off. You can only have the lines that show connections in the cortex, or from the cortex to the subcortex. So that's another way of visualizing lots of data sets together. This is actually a screenshot from a Brain Explorer application. So this is then completely interactive. You can spin it around. Each of those little dots are also a handle to the image. You can click on those and get back to the primary data, so you can see where that data comes from. And then also you can turn on and off experiments, and you can color code it any way you want. So far, all the visualization I've shown is really like per experiment. Given this injection site, where did it go? But because we've voxelized all the data, you can actually transpose all the data to ask the question, which injections come to me? So here, for example, I'm given a planar view of the atlas. I'm going to select this point. And what I'm showing here are all the experiment that has signal going to this point. And then the lines you're seeing here is that tractography line from the injection site to my target point. So what this is really a virtual retrograde experiment, because we are now finding not only do we know from a particular injection where the projection goes to, you can now ask the data in reverse, who comes to me? And if you pick in a different part of the thalamus, in this part here, the green dots represents injection in the cortex. So here, in this part of the thalamus, the only particular part of the cortex projects to this part of the thalamus. And then in this interactive view, you can move the cursor around. And then you can, just by moving the cursor slightly, you can move to a completely different part of the cortex. So from here, you can, in this website, you can actually discover the spatial relationships between two systems and how they're connected to each other. So far I've shown you all the things that you can do on our website. One of the things that we try to do is to make sure all our data is available for analysis. All the image data can be downloaded, and all the gridded data can be downloaded, so that you can construct, for example, a connectivity matrix. This is a typical way people like to show connectivity. So this is shown as each row is one injection, and each column is one of the structures in the right hemisphere and the left hemisphere. This data set was then, in our recent paper, was then modeled so that it became a structure-to-structure matrix. And from those, you can do network analysis that you typically would do to see how close this is to a small world network, if there are hubs, that sort of thing. And because of the spatialness and the spatial mapping of the data, you can also ask questions about topography. So in this rather colorful figure, what we did was we took all the injections in the cortex and then cluster each of these injections together simply by its whole brain projection. And this provided, and then we color-coded in a systematic way, reds up the front, then blue, then the yellow greens, and then the blue, and in the medial part, the purples and the pink. And then we took the center of each cluster and then tried to visualize these 21 different centers together in this composite view. So in this composite view, you don't see. So in this composite view, then each of the 100 micron voxel is drawn as a circle. Because now we're trying to show 21 experiments, that you will get 21 different circles at each voxel. So then the experiment with the largest density, or it's then put at the back, and then it's sorted so that you get the largest density at the back and the smallest one up the front, and everything is perturbed, so all the colors are shown through. So this is trying to give you a sense of what the cortical to subcortical projection. So you see this color layout, the map. There is actually, from this view, you can actually see that in the striatum, that the topography in the cortex is nicely preserved in the striatum. And then it's also nicely preserved in the thalamus. So from the data set, you can actually start to poll at topography of the connection. Well, here I showed it to you as a sort of a visual analysis. Because everything is quantitative and spatial, you can actually compute p-values for these similarities. So we haven't done so, but I know there are many groups who is interested in that, and they can actually grab all our data and do that analysis as well. And we also provide them with that average template, because everything is mapped to that average template. So as long as they've done the image registration to their DTI fMRI data, that analysis can be done. All right, so I'm going to end off to talk about the API. As I said, we have an API to our data, so you can use the website to explore the data, and if you want to do your analysis, you can grab almost everything you see on the website through our API. Our website is built off the API, so anything the website can do, you can do as well. So we welcome mashups of our data. And then in order to facilitate mashup, we have lots of interactive examples, interactive UIs to give you, give you a sense of some of the things that you can do with the data. So there are about 300 people at the Allen Institute. Everybody probably had a hand in creating this data set. I'm calling out basically all the algorithm and visualization developers here. Jan did all the registration, Leonard did all the segmentation, David and Chris did a lot of the visualization, and then this people who do other people do our web application, and then we have different layers of project managers to help all of this thing happen. So here you go.